Hello everyone, welcome to our next review on immune therapies in amyloid light chain amyloidosis. This article, published by Harn et al., talks about immune therapies, the new modalities available for treatment of AL amyloidosis. As we all know, amyloidosis can be categorized into A type, AL type, or ATTR type. Light chain amyloidosis is due to misfolded immunoglobulin light chain secreted by clonal plasma cells. Traditionally, it follows multiple myeloma, and treatment is exactly similar, utilizing a combination of PC targeted agents, that is plasma cell targeted agents. Recent development promotes the clearance of amyloidogenic light chain from affected organs rather than targeting abnormal plasma cells. Toxicity is a main problem in the treatment if we are following the multiple myeloma regimen. Improvements in survival in both multiple myeloma and AL amyloidosis have largely been the result of widespread adoption of novel agents, including proteasome inhibitors, immunomodulatory agents, and anti-CD38 monoclonal antibodies. Here, they've talked about the first immunotherapy to gain approval of FDA for treatment of MM and subsequently AL amyloidosis. Now, malignant plasma cells in MM and AL share several key features, which are relevant to immunotherapeutic approaches, including surface expression of CD38 as well as B cell maturation antigen, along with GPRC5D. These three proteins are the targets of most immunotherapeutic agents developed to date. And daratumumab, which was referred earlier, this one, the anti-CD38 antibody, is already a part of current standard of care first-line treatment for AL amyloidosis. This is in combination with the CyBOD regimen, that is cyclophosphamide, bortezomib, and dexamethasone. The immune-mediated toxicities occur frequently as a result of T cell activation, with both BSA as well as CART. This is chimeric antigen. We had discussed upon chimeric antigen receptor T cell therapy in the earlier video. Please watch the same. Please watch the same. Sorry for the interruption. Please watch the same for further info. Now, these include cytokine release syndrome, a systemic inflammatory response characterized by fever, hypertension, hypoxia. This is a severe adverse effect which must be taken into account. We'll come to that specifically, depending on whichever immunotherapy causes CRS as well as the other adverse effects. Development of CRS is a particular concern in cardiac AL amyloidosis, who often lack sufficient cardiovascular reserve to cope up with this acute cardiocirculatory stress. Now let us go to monoclonal antibodies. We have monoclonal antibodies, antibody drug conjugates, bispecific antibodies, chimeric antigen receptor T cell therapy as the major immunotherapies available for the treatment of this condition. Daratumumab and isatuximab. These are two anti-CD38 antibodies approved for multiple myeloma treatment. They promote cell death by targeting CD38 molecule, and here we can see daratumumab is administered subcutaneously while isatuximab is available as IV. Both have shown effectiveness in treating multiple myeloma, including in patients refractory to other treatments. Both have also been used in the treatment of AL amyloidosis, which is a very much related condition. And they are often used in combination with the CyBOD regimen, which I mentioned earlier. They have favorable safety profiles with manageable side effects. And clinical trials are investigating the use of these antibodies in various settings as well. SLAM F7 receptor is another target for our therapy. Here I'd like to talk about Andromeda trial in which daratumumab was incorporated into standard first-line therapy. Here, in this trial, 338 patients with newly diagnosed AL were used, and either alone or in combination with daratumumab, the therapy was given, and then threefold increase in the rate of hematologic complete response was seen which was the primary endpoint of the trial, which was achieved in 53% of the patients receiving daratumumab as compared to 18% in the control group. Now, as I already told, they've been evaluated in several clinical trials. And here in another trial, we can see that early results have noted high hemorrhagical rates, but there was also high mortality. So whether daratumumab monotherapy or subsequent treatment will lead to lower early mortality rates remains to be seen, and it requires more clinical trials. Elotuzumab is a monoclonal antibody targeting SLAMF7, the receptor which I told about earlier, for treatment of patients with multiple myeloma in combination with immunomodulatory agents and proteasome inhibitors. Now, let us go to antibody drug conjugates. Belantamab mafodotin, BELA in short, is an antibody drug conjugate approved for multiple myeloma but later withdrawn due to a phase 3 trial. Now, BELA is being investigated in AL amyloidosis, showing promising results in small studies. It has a high hematologic and organ response rates in AL patients. Ocular adverse effects, primarily keratopathy, are common. Dose adjustments, dose reductions are often necessary to minimize toxicity. And another ADC targeting CD38, that is STI-6129, is currently under investigations. Let us go through this paper in brief. These are the main points which I told right now. A prospective study investigating BELA at a dose of 2.5 mg per kg is ongoing, and a preliminary report after enrollment of 25 out of planned 36 patients with a median 3 prior lines showed high overall hematologic response rate of 60% and 3-month organ response rate of 20%. Responses were rapid. Ocular adverse events were seen and grade 3 keratopathy was also seen. The rest I've told about already. STI-6129 is an oral ADC consisting of an anti-CD38 antibody linked to MMAF. Now let's go to bispecific antibodies. Mechanism of action: they redirect T cells to target tumor cells. Teclistamab is a BCMA CD3 BSA approved for multiple myeloma. Limited data suggesting promising efficacy of teclistamab in AL amyloidosis is available. It has a high hematologic and organ response rate with respect to efficacy, and severe infections remain a concern, similar to its use in multiple myeloma. Here, cytokine release syndrome have been reported, but they are generally manageable. Infections are common. Rates of CRS were consistent with the previous report, occurring in 4 out of 7 patients. Here they have cited several studies. We can go through the same. The reference will be posted along with the video. This is a very recently published paper showing the new immune therapies. Now, let us come to chimeric antigen receptor T cell therapy. I had discussed this already in the previous video, but then I'll go through the mechanism in brief. CAR T cell therapy basically involves engineering T cells to target tumor antigens. And then the primary target antigen for CAR T cell therapy is BCMA, which I had mentioned earlier. And now, efficacy. With respect to efficacy, CAR T cell therapies have shown promising results in multiple myeloma with high response rates and improved survival. Limited data says that CAR T cell therapy may be effective for AL amyloidosis. Early and late toxicities can occur, including CRS, cytokine response syndrome, cytokine release syndrome, cytopenia, and secondary cancers. Cardiac toxicity, cardiac adverse events have been associated, especially in patients with AL amyloidosis. And there are newer advancements in the same. Late toxicity, prolonged cytopenias, and hypergammaglobulinemia can be seen. Now, the advancements, despite lower expression of BCMA compared to in vivo validation studies confirmed efficient targeting of these cells by HBI0101 and XC201. This is basically a locally developed CAR T product which has shown promising results in AL amyloidosis, including those with advanced cardiac disease. Now, striking efficacy was observed in some studies in which this treatment modality was used. In overall hematologic response rate was 100%. This is to be taken into account. At 30-day post CAR T-cell infusion, 6 of 9 evaluable patients were MRD negative. So, in conclusion, while showing a manageable toxicity profile for frail AL patients, even with excessive cardiac involvement and multiple previous lines of therapy, the immune therapies promise unprecedented rates of hematological responses, which are rapid and deep, including MRD negativity, which is to be kept in mind while treating AL amyloidosis. Hence, we must aim for immune therapies furthermore in the future. That is, they are expected to become a powerful adjunct to anti-plasma cell therapies in the near future. Thank you everyone for your patient listening. See you next week.